It's my pleasure today to introduce my friend and colleague, Steffanie Strathdee, who's going to be talking about From the Bog to the Bedside, the story behind iPath, the first dedicated phage therapy program in North America. You can see all the good stuff there on the left side. Definitely need to check out her book. Next slide. Just about today's speaker, my friend Steph is an infectious disease epidemiologist who co-directs our UCSD Center for Innovative Phage Applications and Therapeutics, iPath, and she also co-directs the international core of our San Diego Center for AIDS Research. She spent the last 2 decades focusing on prevention of infectious diseases, especially in marginalized populations and low-income countries, and has published over 700 peer-reviewed publications. In 2019, she co-authored a memoir, The Perfect Predator, Scientists Race to Save Her Husband from a Deadly Superbug. We're going to hear a little bit about that today. And it basically relays iPath's first experience with phage therapy, and what we're going to be doing going forward is what she's going to tell us about today. And of course, in 2018, Time Magazine named her as one of the 50 most influential people in healthcare, which is really pretty cool. Next slide.
All right. Actually, Steph, we're good to go. I'll pause the share and you can take over.
Okay, great. I am just going to get my slides going here. Just bear with me. The little bar comes across the top and it doesn't. Okay, here we go. All right, so hopefully everybody can see this. This is actually an unusual kind of a lecture because I'm going to be talking about antimicrobial resistance, which is the next pandemic that is already here. But I'm going to be talking about one of the innovative solutions to combat it, which is phage therapy. And I'm going to work this through the lens of a personal story, which affected my family. Some of you are familiar with some aspects of the story, but hopefully something will be new. So first, a couple disclosures. My husband and I never dreamed that we would be working in phage therapy, and we hold stock in a phage company. I will not be speaking about any specific companies today. I'm an unpaid advisor to another phage company. All patient photos today are used with permission, and I also include my land attribution to the Kumeyaay peoples as we are on their unceded land. So this story really began in 2015 when my husband Tom and I were on vacation in Egypt. Tom is also a UC San Diego professor, and we're both HIV prevention researchers. He looked to be in perfect health, except he was overweight. He's about 6'5" and close to 300 pounds in these photos, but you can see that he looked to be in perfect health. He was hamming it up at the Cairo Museum and crawling backwards down into the Red Pyramid outside of Cairo. And that was on Thanksgiving Day of 2015. And yet 3 days later, he was in a local clinic. There was no hospital, seriously ill with what I thought was food poisoning, but which quickly deteriorated into something very different. Here he is being rolled into a CT. And the physicians that were there didn't like the looks of what they saw on this. It turned out that he actually had a gallstone that had blocked his bile duct and caused an abscess to form the size of a small football. And he'd been walking around with this abscess in his abdomen for quite some time. So he really didn't have food poisoning at all. He had a gallstone attack. But I didn't realize this until much later. And unfortunately, that lovely little abscess in his abdomen made a nice little apartment for a nefarious microorganism to move into. We were first medevaced to Germany since he was too weak to be sent home initially. And you can see here that I'm in full PPE. And I did this selfie and posted on Facebook thinking that, we're going to be laughing about this in a few days. And I even joked with him and told him that, he almost saw King Tut's tomb, but he came close to being, feet first in his own tomb. And I didn't realize how close it was to being the truth because he was fading in and out of consciousness at this time. The doctors there removed the gallstone that was blocking his bile duct, and I thought that was going to solve everything. But they said, no. In fact, that's just the beginning of the problem because we've got this putrid fluid inside this abscess, and inside that is growing this organism, and they cultured it. It took a few days to come back, and it was Acinetobacter baumannii. And I remembered plating this organism on my Petri dishes back in the 1980s. And when I I remember telling Davey, Dr. Davey Smith, who introduced me, who's now our division chief at UC San Diego, I mentioned that, Tom had this infection, and Davey said, well, that used to be a really wimpy organism. But over the last couple of decades, it unfortunately has gained incredible superpowers. It is on the list of the WHO's most critical superbugs that are of threat to human health. And so by a superbug, I mean a bacteria that has resistance to multiple antibiotics. And you see this one is on the top of the list. It's considered to be critical, and it's often acquired in hospital and clinic settings like the one that he was in. We'll never know exactly how we acquired it, but we found out later it was an Egyptian strain because it was sequenced. I started to get a little worried about this at this point. And then the doctor said, we're going to run an antibiogram, which is essentially an antibiotic susceptibility profile, to make sure that we are on the right path in terms of giving him different antibiotics. And unfortunately, the antibiogram came back a few days later, and I was given this before I had a chance to talk to any of the health providers. And I didn't understand German, and you might not either, but you don't need to, because when you look at this chart, you see every R in this list stands for resistance. It was only partially sensitive to 3 different antibiotics off the top. And I call these gorillacillins because they have to be infused into the patient, and they are quite toxic. So, for example, colistin is the last resort antibiotic. It was developed in World War II. It's extremely nephrotoxic, and yet it's something that providers cling to because it's one of the last few antibiotics that is used when someone is as seriously ill as Tom and there's nothing else to give them. So I started to get really worried about this point. And then I realized, that Tom was unfortunately the poster child for this post-antibiotic era that we've been hearing that we are entering. In fact, the Centers for Disease Control directors, the last several of them, as well as the head of the UN and the WHO, have said that we are entering a post-antibiotic era. That means that an era where antibiotics are not going to be working anymore, and that simple infections that used to be treatable are no longer treatable. It's estimated that by 2050, 10 million people per year, that's one person every 3 seconds, is going to be dying from a superbug infection, and that this will cost $100 trillion per year to the global economy, mostly through loss of productivity. So I started to read up on this and realized that I had gotten, really behind. I was blindsided by the seriousness of this pandemic that has been a slow burn for the last couple of decades. And in fact, there was a paper that was published in Lancet in early 2022 that estimated the number of people that had died due to antimicrobial resistance in 2019, the year before COVID started. And it estimated that 1.2 million people died directly as a result of AMR, but that about 5 million died with AMR, so where it was a contributing factor. So this is much higher than anyone had ever thought. It's more deaths per year than malaria or HIV or tuberculosis. And yet, why are we in this situation? Well, it isn't just that people are misusing antibiotics and that doctors are prescribing it when it shouldn't be, or that patients are taking it when they shouldn't. In fact, in the US, Canada, and many other countries, 70% or more of antibiotics are actually used in agriculture and in animal husbandry to make animals grow fatter faster, because they act as growth promoters. Even when they're banned to be used as growth promoters, they're often used off-label like this, and there's lots of loopholes. So in fact, one of the more serious antimicrobial resistance genes confers resistance to colistin, that last resort antibiotic that I mentioned that Tom was receiving. And the year and month that Tom fell ill, a paper was published in Lancet reporting the MCR1 gene that confers resistance to colistin. It was identified in China. And this is not surprising because China was using colistin in their pig industry. They were feeding colistin to pigs. And, you know, I don't want to single out China here because many other countries were doing this, but China was one of the last countries to be doing this. And they reported this, and it cast a pall over the whole infectious disease community. I remember people saying, oh my God, this is our last resort antibiotic. What are we going to do if we don't have colistin? Now, China banned colistin use in pigs in May 1st, 2017. And that ban has been associated with declines in the prevalence of MCR1 in China ever since. However, colistin is still being exported from China to lower and middle income countries such as Bangladesh, Pakistan, and Nigeria. And when Tom's organism was sequenced, it was found to include not just the NDM1 gene that was given that awful name, the New Delhi AMR gene, but also this MCR1 gene. And it was a shock to people, but it shouldn't have been a shock because this MCR1 gene was already in 30 countries. So, and we don't want to give the impression that this is just a problem overseas or lower middle income countries, because just in April of this year, bacteria resistant to colistin were found for the first time in Los Angeles County wastewater. So the MCR1 gene was identified there as well. So this is a problem globally because, once an organism obtains antimicrobial resistance genes, its globalization and travel spread these organisms and these genes worldwide. So there's really a big problem when you look at the epidemiology of antimicrobial resistance, because we have very poor surveillance, as indicated in that example. There's a lack of reporting requirements and standardization. The UN and other multinational agencies have promoted national action plans for antimicrobial resistance, and most countries have them, but there's no accountability. There's no milestones, and anybody that's saying, oh, you haven't met your goals. And so there's no teeth to these plans, and that includes the US. There's also powerful lobbying from the pharmaceutical industry and agribusiness that keeps using different antibiotics like streptomycin, which is a medically important antibiotic. It's still being used in citrus around the US and other countries. There's low awareness of antimicrobial resistance among the community and policymakers. And finally, there's very few new antibiotics being developed. So here's just one graph that shows the discovery of new classes of antibiotics over the last number of decades. And as you can see, since 1990, there's no new classes that have been entering the pipeline. Most of the new antibiotics are riffs on older antibiotics. So they just, tweak it a little bit. So why is this happening? Well, pharmaceutical companies are seeing that they're not going to make much money if antimicrobial resistance puts their new product out of business within a couple of years. And antibiotics aren't big money makers like CAR T therapy and things like that. So, so this is a big problem, and it means that we're getting really behind because the microbes are winning. And so back to our story, Tom was medevaced back home to San Diego, where my colleagues were caring for him. And that includes Dr. Davy Smith, who introduced me today, and many other people, some of whom might be watching today. And Tom was really, really sick. I mean, here you can see that he's on a ventilator. That meant his lungs are failing. He was on 3 different pressors or medications to keep his heart working, and his kidneys were hanging on by a thread. And around the time this photo was taken in February of 2016, so he'd been in the hospital about 4 months, I was told that he wasn't going to make it. And I was asked if I wanted to start kidney dialysis, and that was going to be imminent. And I had a conversation with Tom where I asked him if he wanted to live. It was the most awful conversation you could ever imagine having to a spouse. And I didn't know if he could hear me, but I asked him, if you want to live, honey, please squeeze my hand and I'll leave no stone unturned. And I waited and he squeezed my hand and I was elated. I had my little blue gloved fist and I pumped it in the air. And then I thought, oh crap, like, what am I going to do? Right? I'm not a medical doctor. I have some of the best infectious disease physicians in the world caring for him. And they say that they've run out of options. And so I did what anybody would do, what you would do if this were you. I went home and I hit the internet. I used PubMed, the National Library of Medicine search engine that's publicly available. And I put keywords in there like the name of his superbug, Acinetobacter baumannii, multi-drug resistance, and also alternative treatments. And lo and behold, up came a paper that mentions something called bacteriophage therapy, or phage therapy for short. And a little bell rang in my mind because I have a rusty old degree in microbiology from the University of Toronto back in the 1980s. And I learned that phages are viruses that have naturally evolved to attack bacteria. And I never knew that these had ever been used to potentially treat people or animals with bacterial infections. So I started to read up on this very excitedly. And it turns out that phages had been discovered over 100 years ago. And in fact, the earliest report of phage activity was in 1896 by Ernest Hankin, who was a Brit who was studying the Ganges and Jamuna rivers in India. Now, he didn't know what he was looking at when he saw that this water that passed through a Pasteur filter was able to kill cholera bacteria, but he deduced that something was going on. And that kind of experiment was repeated over and over again by Russians and Poles and another Brit, Frederick Twort, who thought maybe that he was looking at a virus or an enzyme. But again, the light microscope was the only available microscope at the time. And he wasn't sure. And it wasn't until Félix de Harel, a self-taught French-Canadian microbiologist in 1917, repeated this same kind of experiment during an outbreak of dysentery in Paris. And when he realized that this filtrate could kill this Shigella, I believe it was, bacteria, he thought we should use this on the children who were dying of dysentery to see if it can cure them. So he first experimented on himself, his family, and his lab mates. Obviously, there was no human subjects committees back then because that kind of self-experimentation would never fly today, but it was very common back then. And when there was no untoward effects, he, with permission of the hospital director and the parents of children, gave this filtrate to children suffering from dysentery, and they were cured within 1 to 2 days. So that was the first time phage therapy was ever administered. That was 1919. So over 100 years ago. And then since then, phage therapy had something of a heyday in the 1920s and 30s. Félix was the inspiration for the Pulitzer Prize winning novel Aerosmith. And he also helped this fellow pictured in the middle, Georgi Eliava, who was from the Republic of Georgia, what is now the Republic of Georgia. He helped Georgie set up what became the first dedicated phage therapy center in the world, what is now called the Eliava Center. And of course, phage therapy is not used standardly in the Western world anymore. And why? Well, part of the reason is that penicillin, which was discovered in 1928, was brought to market because of World War II in the early 1940s. And phage were seen as finicky. They needed to be matched to the specific infection, whereas penicillin was a wonder drug because it had these broad spectrum types of effects. You didn't even need to know which kind of bacterial infection you had. You just give them penicillin. And of course, now we know that having this kind of scorched earth approach to treatment is not a good thing because antibiotics can kill the friendly bacteria in our microbiomes. But nevertheless, the heyday of what became big pharma began, and phage therapy continued to be used in Tbilisi and other parts of the former Soviet Union and in Poland, but fell out of favor in the West. And also part of the reason is that this was seen as Soviet medicine, Soviet science, and the Soviets were the enemy. So this geopolitical bias has hung over the field of phage therapy for decades. And you'll see that's Georgia Liava on the right, pictured with a much older Felix de Harel. And the medical history of this is fascinating, but I was just skimming through all of this, and I just realized, wow, we can't get phage therapy for Tom because it's seen as experimental treatment in the US because clinical trials to show that it have never been done. So in the 1940s, early on, the first electron microscope came to be in Germany, and phages were one of the first organisms that were ever seen. In fact, this is a modern day electron micrograph of a bacterium stained in orange being attacked by phages that are stained in green. And you can see that these ones look a little bit like alien spiders. And now we know that phages are the most populous and oldest organism on the planet. They've been duking it out with bacteria for almost 4 billion years. They come in all shapes and sizes. There's just a few of them that are pictured on this slide. And they need to match to the specific bacteria. They attach to a receptor, and like all viruses, they enter into the bacterial cell. They enter into the cell. This time the host is a bacterial cell. And if it's the lytic cycle, they turn it into a phage manufacturing plant. And these baby phages are made, and when given the kill signal, they burst out, and the bacterial cell dies in lysis. And these virions go on to attack new bacteria. But again, they only attack the bacteria that have the receptor that they match to. So in this way, they don't harm any of the friendly bacteria in our microbiome, and they keep repeating this cycle of lysis, etc., until there's no more bacteria that they match to, and then they're naturally excreted by the body through the reticuloendothelial system. The liver and the spleen play the biggest role in this case. So it's a very self-limiting property that is very interesting and advantageous for phage therapy. So where do you get phages from, and how do you select them? Well, this is kind of the weird part, because wherever you have a lot of bacteria, you have a lot of phages, which are the perfect predator, if you will. And so here we have a flask of sewage, and you can get a Petri dish that's streaked with different bacteria. Here, these little globs are our pal, Acinetobacter baumannii, and you add a drop of your suspension from, say, the sewage, and you incubate it for 24 to 48 hours. And if it comes back looking like this, a bit like Swiss cheese, then that you get really excited. And because that means that these phage are present and they have gobbled up those bacterial colonies. And so you have phage that will kill the bacteria that you want to kill. And so what you can do is pluck out those colonies, and those phage can be added to more bacteria in suspension, and you let them do their thing. And so you grow up more of this suspension, but now you have a lot of this debris. There's the bacterial cell walls that often contain lipopolysaccharide have an endotoxin. And so you've got to purify that suspension before it's given to people or animals. And we'll get back to that later. So I got really excited about this and thought, could we use phage therapy to cure Tom? And I approached Dr. Chip Schooley, who was the head of infectious diseases at UC San Diego at the time. And Tom was looking like this. I mean, he literally was very, very close to death. And Chip said to me, if you can find some phage that are a match for Tom's bacterial isolate, I'll call the FDA and see if I can get permission to give it to him on a compassionate basis. And that meant that Tom was dying, and even Chip was seeing that. So it was a race against time. Now, I wrote total strangers. I found their names on the internet. One of them was Dr. Rai Young from Texas A&M, who heads up the Center for Phage Technology, or he did at the time. He's retired now. He agreed to turn his lab into a command center, and all of the people pictured on the right-hand side of this slide are people that put their work and even their doctoral thesis on hold, looking for phage that would be a match for Tom. And so they took samples from barnyard waste, sewage, soil from, anywhere around. And the woman with the necklace, she's now Dr. Adriana Carolina Hernandez. She found 4 phages that were a match, and we were very excited. In fact, Chip then got on the phone and called the FDA, and he reached Dr. Cara Fiore, who was heading up the unit that was in charge of fecal transplants at the time. And she had never, been involved with any kind of phage therapy in the US. This was unheard of at the time, but she knew all about what phages were. And she said, boy, this is a perfect situation here, because we've been hoping to have a patient who was eligible to get phage therapy because they were close to dying, a family member that was willing to give it to him, a doctor like Chip, who was willing to preside over the protocol, a phage community that was willing to stand up and get involved to find phage to help match to the isolate. And so this was, something that was really remarkable. Now, Dr. Fiore said, now there may be some folks that you haven't contacted yet who might be willing to help. And Chip said, well, Steph's done a pretty thorough search. I think she's reached everybody that's working on this, at least in the US, because we didn't have much time. We weren't going to reach out anywhere else. But she said, no, I don't think you'd know about this group. The US Navy and the US Army are actually working on this. This is Lieutenant Commander Theron Hamilton, who has a PhD in molecular biology, and he headed up the U.S. Navy's Biological Defense Research Directorate. This is actually the unit that was involved in, detecting anthrax in the contaminated letters that were sent as a bioterrorism threat. Actually, I learned later that it was phage from this group that was used to detect the anthrax. They had phage samples phage that had been identified from samples from the bilges of ships all over the world. They had a collection of Acinetobacter baumannii phages that had never been used on people, but they were willing to see if any of these phages were a match for Tom's isolate. So the fellow on the right-hand side of this slide is Dr. Biswajit Biswas, who Theron affectionately dubs the phage whisperer, I kid you not. And he was actually the person who identified 4 phages from the Navy that were a match. Now, you may be wondering, since we found 4 phages already from the Texas group, now mind you, the Texas group had reached out to teams all around the world, and the Belgians and the Swiss and the Indians, and even the folks in Georgia had offered to help. When the Texas team found 4 phages, you might say, well, why did we need any more? Well, the prevailing wisdom at the time was that you really wanted to have as many phages as possible, because you're hoping that the bacteria, if they have multiple phages to respond to in the attack, that they would have a harder time, generating resistance to multiple phages. Of course, what you really want are phages that attack different receptors of the bacteria. And we didn't have the luxury of time to sequence these phages, so we didn't know how different they were. That issue will come up later. But nevertheless, we ended up with 2 phage cocktails. Now, Dr. Schooley at the time had this dilemma, because he'd never done anything like this before. Nobody in the US had, and he didn't know how much phage to administer, what routes of administration would be best, how often and how long. He consulted Dr. Maya Mirabishvili, who was trained at the Iliava Center in Tbilisi, and she said, we don't treat people intravenously with phage therapy because we don't have the ability to measure or remove the endotoxin, but I can tell you how we would administer it through the catheters in Tom's abdomen, where the source of the infection was closest to. So she advised on that aspect. And then Dr. Carl Merrill, shown on the right, he led an intramural program at NIAID that was shut down actually by Tony Fauci in the 1990s. And Carl said, look, this guy is systemically infected. I mean, you need to treat him intravenously, because if there's a hidden reservoir of bacteria that the phage can't reach, then those bacteria could become resistant to the phage very easily. And so Chip mentioned this to me. I agreed that we should treat him intravenously. And Chip said, well, let's treat him both. We'll go through the catheters originally. And if he survives that, then we'll go intravenous. And so we knew that we were taking incredible risks, but or we thought we were. Nobody really knew. But Tom was going to die otherwise. Now we had one final problem to deal with, and that was this endotoxin issue, because the FDA, even though they didn't have any guidelines for phage therapy, they know about endotoxin, and their metric was that you should try to get the endotoxin down to 5 endotoxin units per kilogram per hour of body weight of the patient. And we were way over that, but the Texans had already sent their phage preparations to San Diego to treat Tom. And then they did these endotoxin assays and realized that it was way, way higher than it should be. And so luckily, we found out that there was a team of researchers at San Diego State University, our own backyard, who were studying actually phage in marine life, especially corals. But they had an endotoxin removal assay all ready to go, and they agreed to help. So Dr. Jeremy Barr was a postdoc working in the lab of Dr. Forrest Rauer and Dr. Anka Segal, and he worked through the weekend to get the endotoxin level below the threshold. So that was really scary because Tom right now was really clinging to life. We got this phage cocktail, though, with an approval from the FDA through an emergency investigative new drug on March 15th. So this was only 3 weeks from my first email to when we had this phage cocktail ready, which is pretty amazing. And this is Dr. Chip Schooley and Dr. Randy Taplitz, both of whom were instrumental in Tom's case. And they're smiling for the camera right before phage therapy began, because they knew that they were making medical history one way or the other, whether the patient lived or died. So this is Dr. Andrew Purcell. He is an interventional radiologist. And so the phage preparations from Texas were ready first. Those were instilled through the catheters in Tom's abdomen. And when no septic shock occurred, 3 days later, the phages from the Navy were ready. And that's Dr. Melanie McCauley, who was an ID resident at the time. And she was the one who infused those phage into Tom's PICC line, and we just prayed that he didn't undergo septic shock. And 3 days later, believe it or not, even though Tom had been in multi-stage organ failure, and I had to sign the consent form for kidney dialysis the day we started phage therapy, but we didn't need it because Tom woke up, opened his eyes, lifted his head off the pillow, and kissed his daughter's hand, who was on shift with him at that time. Now, everybody in the ICU at UCSD's Thornton Hospital was in shock and cheering and crying, and it was a really remarkable day. Tom left the hospital several months later. He was very deconditioned. He lost over 100 pounds, needed to learn how to walk, how to talk, everything all over again. But his journey to recovery had begun. Now, a couple months later, you can see him. He's looking a lot better. He met Lieutenant Theron Hamilton, who came out to visit him. And Tom's case report was published by Chip and others in AAC in 2017. And that included the protocol for how to do phage therapy, and has been cited thousands of times by now. And this is another scanning electron micrograph I wanted to show you, but it's a special one because it's Tom's bacterial isolate being attacked by the Navy phages. And this SEM was actually generated by the Department of Homeland Security. So I still like to throw darts at this every once in a while, but it's a pretty cool photo, and it's actually included in the book. So our story was presented at the 100th anniversary of the discovery of the bacteriophage, 1 year after Tom was cured. And so this is April of 2017. And after that, the story went viral, but in a good way. So it was picked up by JAMA, who did a Q&A with Chip. Lancet commissioned a commentary, but it was also in Time Magazine, People. We were on the Today Show. It just went on. It was like covered all over the world. And of course, other patients and their families heard this story and they wanted phage therapy too. And because a global village had stepped up to save my husband's life, I certainly wasn't going to turn them away. So we reinvented the wheel, approached some of the same partners who'd helped in Tom's case, as well as new labs, some of whom were even in industry. And we saved some lives, and it was too late to save others, but we've been doing this ever since. We saved enough lives to impress our Chancellor Pradeep Khosla, who gave us seed funding to open what became the first dedicated phage therapy center in North America, the Center for Innovative Phage Applications and Therapeutics. It's a nonprofit. It's based here at UCSD. And the day we opened in June of 2018, Science Magazine published a commentary where a microbiologist was cited as saying, this is a game changer in the field. And since then, there's been lots of case reports and case series that have been published by our team and other teams around the world with some success, and enough success that many countries have started their own phage therapy programs. But it isn't all roses, because this is a very challenging problem, because we get so many requests for phage therapy. There's been over 1,800, and the majority of these folks are not clinically indicated for phage therapy because they still have antibiotic options left. So, and these cases have to be approved on a case-by-case basis by the FDA right now, while the clinical trials that examine whether or not phage therapy is efficacious are conducted. So we have determined through our physicians, Dr. Simon Aslam, Dr. Chip Schooley, and others, that 365 of these 1,800 or so cases are indicated, for phage therapy. And we've indicated, we've initiated phage hunts in 302 cases. Sometimes the patient dies before we even get to this point. And you see that in about half of these cases, we've found lytic phage that will kill the bacteria. But again, there have been other barriers before we can sometimes treat the patient. These are seriously, critically ill patients, some of whom are as sick or as sicker than Tom. And sometimes the patient dies before we can reach them. There's been regulatory or financial barriers. And so we've treated only a minority, about 16% of those where phage is recommended by physicians. So this model is clearly not scalable, but we do have a very clear idea of what we need to do. And I'll talk to you about how we could bring this model to scale. But before I do that, I'll just show you that we are treating a number of different kinds of clinical indications and a number of different multi-drug resistant organisms. One of the most common is Pseudomonas aeruginosa. That organism has been implicated in a recent outbreak of contaminated eye drops. And Dr. David Pride's lab, who leads our IPATH lab, has found phage that are a match for those isolates. So the CDC has said that they will help us treat any patient who has that infection. But there's lots of other types of conditions that that Pseudomonas aeruginosa is associated with as well. The others are Staph aureus, mycobacterial infections, E. coli, and I put this last one, Borrelia burgdorferi, on here because that's the causative agent associated with Lyme disease. That's one of our most common requests. However, for reasons that we still don't understand in microbial ecology, sometimes the phage that we identify or that other labs identify are not lytic phage. They don't kill the bacteria. They're what's called temperate phage. Those phage undergo the process of lysogeny. That means they integrate their DNA into the bacterial cell DNA and hit the snooze button. And those often carry AMR genes or toxin genes, and they're not ideal for phage therapy. It doesn't mean that they can't be used, but they'll likely need to be genetically manipulated. So, that's part of what needs to be done in the future. So what we really think that we need, however, is to build a phage library. And a phage library is needed because right now it's like having a million locks scattered around the world. world and a million keys scattered around the world. And you need to find the keys that match the locks. And so if somebody contacts me and they say, you know, I have a patient with a multi-drug resistant Burkholderia cepacia, I know, okay, there's some folks at Texas A&M who have phage for that. There's some folks in Israel who have some good phage for that. And there's some folks in Alberta who have some good phage for that. And so it becomes this race against time to send the bacterial isolate to these labs if they're willing to undergo a phage hunt, and then to identify those phage and to perhaps manipulate those phages to get them ready in time, all to save the patient's life. But if we had a phage library, even if it wasn't located all in one place, that was, you know, organized where we knew where the phage were and were able to access phage quickly enough, we would be able to bring this model to scale. The Economist and many other outlets have written stories about phage therapy recently, and I just wanted to show you that clinical trials are beginning. Tom's case was kind of this strange moment in the history of phage therapy that helped bring phage back to the West. And as you can see, there's a lot of clinical trials that are now registered at clinicaltrials.gov that are underway. One of these is supported by NIH through the Antimicrobial Resistance Leadership Group. And in fact, IPATH is a site, and there's 19 other sites around the US that are participating in this. And it's for cystic fibrosis patients that have chronic Pseudomonas aeruginosa. And so we are recruiting. So if you know anybody who might be eligible, please reach out. We definitely want to ensure that this translational trial continues. And one of the other advances in the field recently is that the first genetically modified phage cocktail to be successfully used in a human was published in May of 2019. In Nature Medicine. This was a young girl with cystic fibrosis who'd had a double lung transplant and was her new lungs were being attacked by Mycobacterium abscessus. And also she had a disseminated case. So you can see that there's skin nodules, there was liver involvement, and she was actually in hospice. And her mother heard about Tom's case, reached out to her doctor who reached across the pond. And we also got involved. Dr. Schooley advised on the clinical treatment because a library of mycobacteriophages exists at the University of Pittsburgh. It's through an educational program called C-phages, where undergraduates learn how to match phage to bacteria. They get to name their phages if they're brand new. They learn how to sequence them. And nobody ever dreamed that there was therapeutic potential with these phages. So this, for the first time that phages from this library were used, only one was lytic, however. And of course, you want to use a phage cocktail for the reasons I explained earlier. So 2 temperate phages were identified from this library of over 10,000 mycobacteriophages, but they were temperate. So they needed to be genetically modified. So Dr. Hatfull's lab at University of Pittsburgh used a predecessor to CRISPR-Cas gene editing called recombinering to splice out, to cut out the repressor gene and force those 2 temperate phage to become lytic. Now, luckily, the UK government did not see this phage cocktail as being a GMO and allowed it to be used. And Isabel left the hospital within a week. She lived another 3.5 years before succumbing to other underlying problems associated with cystic fibrosis. But her case is even more important than Tom's, in my view, because it's a new era of genetically modified and even synthetic phage that's on the horizon. And my colleague, Dr. Vivek Mutalik, who's in the audience today, is working in that area, as are many other pharmas and biotechs now. Before I end, I want to say that antimicrobial resistance isn't just a problem that has been on a slow simmer. It's gotten worse under the COVID pandemic. There has been a spike in antimicrobial resistance, and not just in hospitals, but in the community. And because of secondary infections that are bacterial infections that follow COVID, but also that antibiotic stewardship has really fallen by the wayside because all resources were being directed towards COVID. So this is a big problem. It's getting worse. And also with climate change and global warming in particular, we see that antimicrobial resistance and climate change are two problems that are making each other worse. And of course, when you have warm environments, bacteria multiply more readily. So that's one reason I wanted all of you to be aware of how serious this problem is and why we need to do something about it. And lastly, I'll just say that there are a number of other phage applications other than the medical applications that I've described today. Phage can be applied in a One Health model, which is the intersection of humans, animals, and the environment. They can be used to disinfect wastewater. They can be used as prophylaxis during outbreaks like cholera, and maybe even to groom the microbiome. So I'm going to pause here for questions. I want to thank a number of our collaborators that have helped IPATH, but of course I have to also personally thank a global village of people who stepped up to the plate to save my husband's life, especially the folks at UCSD, but also around the world, who really put the word kind in humankind. And we realize how privileged we are, because a lot of people in lower middle income countries don't have the resources that we had to get the help we had. So that's why my husband and I wrote our book, The Perfect Predator, A Scientist's Race to Save Her Husband from a Deadly Superbug. So if you're interested in that, feel free to reach out or to ask me any questions here or through email or Twitter, where I'm very active. So thank you very much. And I'll see if there's time for questions.
That's great. Thank you so much, Steffanie. So as far as questions go, please feel free to speak up. If you prefer to put them in the chat, that's fine, and we'll read them out as well. So please don't hesitate.
Hi, Marcel Freire from JCVI. Thank you so much for a wonderful presentation, but also such an interesting personal story. In terms of the matching, I'm not so familiar. How does the process work? How long does it take? Can you talk more about the matching process between phage and when you said they found 4 match?
Sure.
Just curious more about that.
Okay, so what I showed you with the big flask and the big Petri dish was kind of like, very crude. That is the plaque assay, and that's been in existence for, decades. And essentially that's what's done, but it's often multiplexed, with a 96 wells of tiny little wells. And so a drop of suspension from, wastewater or from soil that's been, added to a drop of water, because there can be like a trillion phages in a drop of water. So you add it to the well, and then you incubate it for 24 to 48 hours is all it takes. And obviously, we can't visualize phage because they're 100 times smaller than bacteria. But when you see those plaques, that's a sign that the phage are there and that you have a match. So you may see, certain samples that have those plaques, and you may see some that don't. And the plaques look different depending on the organism. I know Stenotrophomonas, for example, the plaques are absolutely huge. But some folks actually on this seminar today in the audience do a lot of plaque assays. So I know Kazia and Vivek are probably a lot more experienced than myself, but that gives you at least an idea. So it's pretty quick.
Thank you.
Dr. Buxbaum?
My question are several actually. What's the current cost per patient? And in your view, what's the feasible target cost to make this readily available on a much larger scale? So it took us, 50 years to get to the $1,000 genome, and now we're down to the $100 genome.
Yeah.
Do you have some sense of what the dollar figures are for this phenomenon?
That's a very good question, because right now, it's very hard to tell, because any experimental treatment you can't charge for it. So you can charge for the phage hunt or the reagents or the shipping, but you can't charge for the therapy. So basically iPath has been, facilitating phage therapy for free. Now, of course, there are companies out there that are trying to turn this into a business. And I've heard different numbers being tossed around, like $10,000, but at this point, it's anybody's guess, because without having a phage library, and some companies have their own, it can be very arduous to identify phages if you have to send your isolate to multiple labs. But there are some new approaches. For example, now if we just had the sequence of the bacterial isolate, and that could be sent so that a phage could be generated to match that, then that would obviate the need to send the isolate and the timing and the shipping and all of that. So I'm not sure, if anybody on the call has on the seminar has a point of view around this, but it's been something that's been debated, and it's obviously going to change. But I'm certainly one person that does not want to see $100,000 per patient being charged, because with the majority of people that have multi-drug resistant infections dying in lower and middle income countries, that would price phage therapy outside of their reach for sure.
Steph, it's Greg. Just to offer the European slant on this, they're trying to have it on the European Pharmacopoeia, which will then allow for reimbursement through insurance companies, and we'll hear more about that in the weeks to come.
Great. And just so folks know, Dr. Greg German is actually the head of Phage Canada, the newly launched national program in Canada that I'm Canadian, so I'm a flag waver here. But Canada has treated its first 2 patients with a 3rd patient in the wings. And now there's phage therapy programs in Belgium, France, Sweden. There's a phage Australia. Portugal just treated its first few patients. There's a phage UK. Ireland just treated its first patient a couple months ago. So this really is happening, and the geopolitical bias that has hung over this field has started to lift.
So, Davey, I know you're very familiar with Steffanie and this subject, but I just wanted to know if you had any new question or any comments about today's talk.
No, I'm really really happy with the talk. So. Thanks, Steph. Thanks, everybody, for joining. Anybody else? Anything?
All right. If there's no further questions.
When's the movie coming out?
Oh, that is a good
question, Mary.
Well, we have interest from Hollywood. I'm not allowed to say anything other than the fact that there is a screenplay that is in the works, but there is a Hollywood writer's strike right now, as you'll know, and there was a COVID pandemic that slowed Hollywood down for a couple of years. So these are a slow process, but our goal is to use our story as a vehicle to raise awareness about AMR and phage therapy. And that would be one way to do it. It feels a bit surreal, but I'll keep you all posted.
That's great. That's very exciting. I'm just going to quickly share my screen so we can see who our next speaker is for the series. All right. All right, so thank you all for attending today. We're very appreciative for you being here and for Dr. Strahddee's great talk. So next month, we'll have Dr. John Ayers speaking on Tuesday, July 25th from 12:30 to 1:30, and should be very interesting. He's going to be speaking partly about ChatGPT, and it's an area that Davey's very familiar with from a recent paper. So please do attend that if you can. So outside of that, thank you all for coming. Have a great weekend, a wonderful 4th of July, and we hope to see you all next month. Thank you.
Thank you. Bye-bye.
Thanks, Steph. Appreciate it. It's great.